In this video, continuation of acute inflammation, already that vascular event, five different types of that vascular event, that is vasoconstriction, vasodilatation, then increase local hydrostatic pressure, vascular permeability, that means protein as well as fluid level increases that causes edema. Last one is blood status, that is slowing process we discussed. In this one we want to look at six important type of cellular event, totally 11, already 5 vascular event completed. Now 6 different type of that cellular event: margination, rolling, addition, transmigration, hemostasis, and phagocytosis. We want to discuss one by one in details. So that vascular 5 event, who will prove this stuff? So who will prove that 5 steps: vasoconstriction, vasodilation, hydrostatic pressure, vascular permeability, and blood status. So who proved that 6 steps? That scientist name is called as Louis. Okay, scientist name is called as Louis experiment. There are some mechanism. So that experiment is known as Louis experiment. This experiment done by Louis. That's why it have that name, Louis experiment. So here what he did, take pen or pencil, okay, in that forearm by stroking a blunt point, okay, a blunt point, stroking a blunt point object, injury there, so it causes injury here, it is the physical injury. So you note three things here. First one is the line become red in color, that line become red in color, this is the red line. Second, after that area around the stroke become red, after that area become red, it is known as flare. This one name is called as flare. Third, whole area swollen, that name is called as wheal. This is known as triple response. This process name is called as triple response. Why this red line? Because of vasodilation. Why this that flare? Because of that dilated process, because of vasodilation process. Third one, because of here, why that swelling will happen? Because of edema, increase hydrostatic pressure, as well as vascular permeability. So that reason only three things will happen: red line, flare, and wheal. So this one name is called as triple response. This experiment done by Louis, so it have that name, Louis experiment. So what is cellular event? The main functions of that increased vascular permeability and status is to deliver leukocytes at the site of injury. So it having 6 steps. Okay, so first step name is called as margination. What is margination? According to Bernoulli's theorem, okay, that heavier one travel center, lighter one travel periphery. So RBC, WBC, platelet, and plasma, like this way only it can travel. What is the role? Send by using that vascular event we taken WBC out. So first step, taking WBC from center to periphery. This one is the periphery region we want to take. That process name is called as margination. So like a tire and road, who is that tire? This WBC act as a tire, that endothelial cell act as a road. So here traveling, tire on a road. So WBC leaving at the center, WBC leaving at the center and reach to that margin. This process name is called as margination. After just below the gap it stop, okay, below that gap here it stop. So here addition process will occur. So this one will attached here. So this process name is called as addition. Now ultimately WBC will come out. Okay, after attachment only it will come out from that intracellular space to here skin is presented. Okay, so here only bacterium is presented. So here ultimately WBC will come out from intracellular space to extracellular space. This is known as transmigration. This process is also called as diapidiasis. Diapidiasis. So first one is uh, transmigration. Okay, first one is margination, then rolling, third one is addition, then when it will come to out, that one name is called as transmigration or diapidiasis. Diapidiasis. This margination process have another name is called as fermenting. So that margination process having another name is called fermenting. Now this WBC reached to outside. That process name is called as transmigration. It is a 3D space. How WBC count that find out bacteria? Bacteria secrete. So now it reached to outside. So now how it will find out that bacteria secreting some chemicals. That chemicals attract WBC. That chemical attract that WBC. This process name is called as hemostasis. So that process name is called as hemostasis. Movement towards chemicals. Movement towards chemicals. So WBC will move towards to that chemicals. Where that chemical will come, that route, that WBC will move there. After that, phagocytosis process will occur. Lastly, WBC phagocyting the bacterium. So first one is center part. Through migration it will reach to that periphery. Then before that cap here attachment, that process name is called as addition. Then it will move to outside. Okay, intravascular to extravascular, that name is called as transmigration. Now here WBC is presented. Bacteria secreted chemicals towards that chemical only. This WBC will move. That process name is called as hemostasis. After that phagocytic process will occur. So WBC phagocyting that bacterium. Enumerate that 6 steps. WBC at center, then it comes to that periphery. That process name is called as margination. After rolling just before that cap here, that attachment, that name is called as addition. After that it will come out. That process name is called as transmigration. WBC move towards that bacterium. Okay, that name is called as hemostasis. Lastly, phagocytosis process. So first step, that margination, axial blood flow to reverse axial blood flow. How rolling, addition, transmigration together takes place. How WBC rolling on that periphery with the help of receptor. How it will move with the help of receptor. That bacterial cell, okay, that cells having corresponding receptor, and that receptor also presented in that wall of blood vessels. So the WBC also having receptor, that periphery also having receptor. Okay, so that cell surface, this cell surface having receptor, corresponding receptor. Okay, like that endothelial cell, that wall of that blood vessels also having receptor. So interaction of this receptor, the rolling takes place. So because of here WBC also having receptor, endothelium having also receptor, both are having interaction, so bonding. Okay, so interaction of that receptor, after that only rolling process takes place. So this one is the cell, it having receptor. This one is the periphery, this one having receptor. So here bonding will occur. After that only rolling will happen. This way only rolling takes place for addition bonding. So here addition, after that only here addition, then bonding here stops, then it will move to transmigration. Name of that, now we know cells also having receptor, that endothelial cell also having receptor. Now what is the mechanism, what is the receptor name? Okay, name of that corresponding receptor. Complementary addition molecule, CAM. Okay, CAM. That attachment of leukocytes to endothelial cells is mediated by receptor. That name is called as complementary addition molecules. So two type of, we know two type of cell. One is WBC, second one is endothelial cell. Both are having receptors. So during both WBC and endothelial cell, okay, bonding will occur. After that only rolling process will occur. So WBC, what are the receptor? Then endothelial cell, what are the receptor is presented? Both are having 6 receptors. That means complementary addition molecules, CAMP molecules. 6 fair. First one is endothelial molecule. Okay, it acts as a road. Leukocytes, WBC act as a tire. So here it having B-selectin. Here leukocytes, the tire having silyl Lewis X. This one is the receptor. Then E-selectin, silyl Lewis X. What is the role? Rolling. This endothelial cell having E-selectin and silyl Lewis X. Both rolling and addition. Then third one is leukocytes having L-selectin. This one having CAMP1. It having another name CD4. This role is rolling. Then this endothelial molecule having CAMP molecules ICAMP and VCAMP. ICAM means intracellular, VCAM means vascular addition molecules. Okay, so this one having receptor here, WBC having that receptor CAMP molecules B2 integrin, B1 integrin. Here both receptor roles addition. After that now now it addition process will occur, then it want to go out. So here both together having WBC and WBC and endothelial cell having single receptor only, same name only, that is BCAM1, or it having name CD31. So by using these two receptor only, transmigration process will occur. So endothelial cell, first one having B-selectin, second one having E-selectin, third one having CAMP1, it having another name CD34, fourth receptor ICAMP1, fifth receptor VCAMP1. Okay, then here the WBC having, first one having silyl Lewis X, then silyl Lewis X. Third one is L-selectin, B2 selectin, B1 is a B2 integrin, then B1 integrin. Okay, after that both are having same receptor, name is called as PECAM1 CD31. So by using this receptor here, rolling, then addition, then transmigration process will occur. Transmigration, now it will reach to outside. After that bacteria will secreted chemicals. Okay, so bacteria now secreted that chemicals. That process name is called as hemotaxis. Bacteria, okay, it is secreted chemical, that chemical attracting the WBC. This one is the unidirectional motion. What are the four chemicals? It will secreted four type of chemicals. What are the four chemical? Leucoderine B4. Then complement system, it is secreted two chemicals C5A and 3A. Cytokine, that secreted interleukin 8. So these are the four chemical. Leucoderine B4, complement system 5A and 3A, interleukin 8. So these are the chemicals secreted by bacterium. After that, what is the process? Final one, phagocytosis process will occur. So this is all about vascular and cellular event of acute inflammation.